In this video, we're going to take a closer look at the life cycle of conifers, and we're using this as a model for gymnosperms. And like all plants, they exhibit the alternation of generations. Which includes sporophyte generations and gametophyte generations. So to start, conifers and gymnosperms in general are heterosporous. Remember, hetero means other or different. And so what this means is that there are separate male and female spores that are produced in the process. Now they are also monoecious, and mono means one. So it means that the male and female, and this is the part that's heterosporous, so we've got male versus female, but they're on the same plant, which gives us the monoecious. So you can see on this picture here, we have so the cones here, I should say, these are the structures that contain the gametophytes and the reproductive structures of the tree. And you can see that they're separate or different female cones versus male cones. And we'll talk a little bit about the difference on the following slides. First of all, let's take a look at the female structures. So the female cones are known as ovulate cones, and they are larger strabili. So strabili is just kind of a fancy word for cone. And they're made up of scales. So each one of these is a scale. And each scale is a sporophyll. And if you remember from, I think it's when we talked about seedless vascular plants maybe, a sporophyll is just a modified leaf that produces spores. So you've probably seen a pine cone before, and you probably wouldn't call it a leaf or full of leaves, but these are actually modified leaves. Now each scale has two megasporangia, and megasporangia are just kind of the female structures that are going to produce the female gametophyte. So each megasporangium consists of an ovule surrounded by a kind of an integument or a coating of cells. And so here it doesn't, in this picture, it doesn't show, that much detail, but you can see the ovule. And so this ovule is essentially the megasporangium here. And within this ovule, there are megasporocytes that are ultimately going to form megaspores. So remember that the megasporocytes are inside the ovule here. And these megasporocytes are diploid. So remember, they've got two copies of chromosomes. And meiosis is going to happen, so that diploid condition is going to be changed to a haploid condition. And so ultimately there are going to be formed 4 haploid female megaspores. And they're showing a megaspore right here. Now, out of these 4 megaspores that develop, 3 of them degenerate. So 3 of them are essentially useless. But one of them survives, and it's usually the kind of the bigger, stronger one. And that one is going to go through mitosis. And remember, it is haploid, so mitosis is just going to produce more cells that are also haploid. And so ultimately, this surviving megaspore is going to form a multicellular haploid female gametophyte. Now, each one of these ovules, so each one of these kind of openings right here, has an opening near the base of the sporophyll. So in this case, it doesn't have it kind of labeled on here, but this opening right here, pretend like the pollen's not here yet. And this is called the micropyle. And it's interesting because this micropyle develops a sticky pollination drop. And So ultimately what's going to happen is that when pollen from the male plants is released, it's going to be trapped by that sticky pollination drop, and it's going to be pulled into the ovule in order to allow the pollen to fertilize the egg. All right, so the micropyle is the opening here, and then after the pollen lands on it, ultimately a tube is going to be formed that leads to the ovule. Now, if we look at the male structures, the pollen cones are smaller, but they are still made up of scales like the female ones are. So each one of these is a scale. And remember that each scale is a sporophyll, which is just a modified leaf that produces spores. And each scale is going to have two microsporangia, which are also called pollen sacs. And these are structures that contain microsporocytes. And these microsporocytes are ultimately going to go through meiosis in order to form microspores. So in each one of those scales, there are going to be two microsporangia. And within the microsporangia, you're going to have these microsporocytes. And these microsporocytes are going to go through meiosis, and so their diploid condition, they're going to go from being a diploid cell to making haploid cells. And so 4 haploid male microspores are going to be produced. Those microspores are going to go through mitosis, and so they are haploid cells that are going to produce more haploid cells. And they are ultimately going to produce a pollen grain. And the pollen grain is what is considered the mature male gametophyte. And so this is the gametophyte of the gametophyte generation in this alternation of generations. So remember, it's the microsporocytes that are making the microspores. So we're going to kind of take one of these and kind of pop it out here and take a look at what's going on in a bigger view. So each pollen grain, and that's what I'm going to draw here, consists of a tough coating, and this is the pollen coat. And it just helps to protect what is inside this piece of pollen. Inside this pollen, there are 2 cells. And each cell has a haploid nucleus in it. Now, one cell is going to be called the vegetative cell, which is also sometimes called the tube cell, because it is ultimately going to form the pollen tube when pollination happens. The other cell is a reproductive cell, because it is ultimately going to fertilize the egg of the female gametophyte. So recall on the previous slide, we said that each pollen grain, so I'll write green here, so one pollen grain, consists of a haploid vegetative cell, which is also called the tube cell, a haploid reproductive cell, which is ultimately going to fertilize the female gamete, and it's encased in this tough tissue that's called a pollen coat. Now, that pollen gets released from the megasporangium, which is located inside those cones. So you can see the cones in the picture here. These are those pollen cones. And you can see this person, they're just kind of moving the plant with their hand. And then all of this kind of cloudy stuff coming off of it is the pollen. And this pollen is carried by the wind in order to go fertilize the female cones that are on other plants. Now, you may be familiar with this kind of pollinating that happens typically in the spring. If you've ever spent a spring in South Carolina, you have definitely experienced it. The air can actually look kind of hazy because there's so much pollen in it. And then that pollen tends to kind of rest on stuff. And so your car is probably covered in pollen, which is essentially plant sperm. So now let's talk about what happens during pollination. And it's important to note that pollination and fertilization are two different things. So don't get them confused. So pollination is just caused by pollen landing on a female structure. So in this case, if that pollen reaches a female cone, it's going to stick to the pollination drop that's at the micropyle. And remember, this picture unfortunately isn't very detailed, but the micropyle is here, is this kind of opening. And before, here it's actually showing a pollen grain already attached. But essentially what the female structure is going to do is it's going to produce this pollen drop that's kind of coming out of the micropyle. So imagine if there's kind of a drop of liquid here. And this liquid is sticky. So when the pollen is working its way through the air, it might land on that pollen drop and get stuck. That pollen drop is then kind of sucked into the female gametophyte in order to basically pull the pollen kind of into the structure. Now remember that the pollen had those two different cells in it. One is the vegetative or tube cell, and that actually produces this tube, this pollination tube, that makes its way to the ovule. The reproductive cell goes through mitosis to form sperm, and these sperm are then going to make their way down this pollen tube in order to make its way to the ovule so it can fertilize the egg. So with pollination, just to recap, the pollen is sticking to this pollination drop where it then gets kind of pulled into the female structure. The vegetative cell of the pollen forms a tube that makes its way towards the ovule. The reproductive cell, and remember these are the cells that are in the pollen, is then going to go through mitosis to ultimately form some sperm. And then that sperm is going to travel down this pollen tube towards the ovule. And the ovule is where the egg is hanging out. And that egg is waiting to be fertilized. So fertilization refers to the fusion of two haploid nuclei. So essentially a haploid sperm nucleus unites or fuses with a haploid egg nucleus. That is called fertilization. I'm sorry, I put it twice here. And then ultimately what that's going to do is form a unicellular, so a single cell diploid, because remember we took one haploid plus a haploid and we get a diploid. And this is forming a zygote. And so the zygote is essentially the first cell with a diploid nucleus. And this is the fertilized egg. Now that cell is going to go through mitosis to just start forming more diploid cells. And this ball of cells being produced by mitosis is called the embryo. Now that embryo is eventually going to be encased in a seed coat, which helps to protect the the embryo from environmental factors. And that seed coat is developed from some of the female tissues. And interestingly, this is a pretty slow process. So seed development can take up to 2 years in conifers. So last but not least, so we've got these seeds with the seed coat, and inside the seed coat is the developing embryo. Those seeds then get released from the female ovulate cones, where they are then carried to other places in the environment. And they might be carried by the wind or water or by animals. It just depends. And if they ultimately end up in an environment that is favorable, germination will occur. And germination just refers to mitosis occurring. So this embryo is just going through mitosis again and again and again. And ultimately, those cells are going to start differentiating until they form the sporophyte. Remember that in conifers, the sporophyte is this tree. So basically, the whole tree is the sporophyte. And then the cones on the tree contain the gametophyte.